Hi friends, you're back with me, your friend for biology, Professor Girish Kukreja. From last few days, we have been actually talking about the PYQs. I don't know, students love a lot PYQs, the previously year questions. I don't know why they love it. But then I thought that if everyone is doing that, let me also do that. So today we'll be talking about some of the questions which have been asked in our earlier NEET exams. Maybe in the comment section you can tell me ki I have been referring to which paper of your NEET exam, which year, right? So which of the following, I'm not wasting much of your time here, so which of the following can be used as a biological control agent for the treatment of plant disease, right? So biocontrol agent which is going to help to protect your plant from diseases, right? So that is a biocontrol agent, right? So here we have lactobacillus, which is going to produce curd for you. Maybe it will help you to what you call as keep your intestine health good, but it's not with the doing anything with the plant disease. Yes, we have the fungi trichoderma. Yeah, so this trichoderma is a fungus which helps the plant to fight against those plant pathogens, especially those which are coming from the roots and all, right? So yes, here you are with the answer. So it is trichoderma which is being used as a biocontrol agent. You have many bacteria like Bacillus thuringiensis. You have viruses like baculoviruses. One of the famous groups there is your nucleopolyhedrovirus, right? Anyways, chlorella, green algae used as food, right? Also for many experiments. Remember, many experiments of photosynthesis done with chlorella. Anabaena, along with its sister nostoc, used for nitrogen fixation. These are cyanobacteria. Remember, they are prokaryotes. So you have, this is also prokaryote to mention. So lactobacillus is a prokaryote, Anabaena is a prokaryote, not cholera, it is chlorella, that is a eukaryote. Coming to the answer, it is trichoderma. Yes, so trichoderma is a fungi which is used as a biocontrol agent. Yeah, next one, the respiratory quotient for tripalmitin. Tripalmitin, triglyceride having one glycerol and 3 fatty acids. Not going much into detail, but then you have that formula in your book where tripalmitin requires around 145 of your oxygen, releasing around 102 of your CO2, right? So amount of CO2 released upon amount of the oxygen consumed, right? So you have 102 by 145. My mathematics is not good, but I have learned the answer. So it is 0.7, right? Maybe because it's going to be less than 1, surely not going to be 0.9. It's going to be near 1, so it is not going to be this, not going to be this, so it is 0.7. At least I know that much logic. So for tripalmitin, it is 0.7. Yes, for proteins it is 0.9, and for carbohydrates it is 1. Yeah, so carbohydrates 1, for proteins 0.9, and for these fats it is 0.7. The respiratory quotient, RQ, amount of CO2 released upon amount of oxygen consumed. Yeah, you know that. Placentation, in which ovules develop from the inner wall of the ovary or in the peripheral part. Yeah, so where you have these ovules, right? Yeah, from the sideways. Yeah. So this is what is referred to as your, I couldn't see the ovules there. But then yes, you have these, the ones which are coming from the inner wall there, or from the periphery, from the sides they are coming. So yes, that is what is called as your parietal, right? I'm not going more detail of this placentation because I have a complete video on explaining all the placentations along with example. You can find the link in the description section for the complete video on placentation. So I'm not explaining that. Persistent nucellus, right? So nucellus actually should go away when the fertilization takes place, but if it still remains, so nucellus, if it still remains in the seed, where it remains in the beet. Yeah, it remains in black pepper, right? So in black pepper and in beet, right, the perisperm, the persistent nucellus is what is called as your perisperm, peristent, persistence, perisperm, persistent, right? So one which remains, it is your perisperm. We know that the stegmen is that inner wall of that seed. Chalaza is the wall of the, what you call as the opposite end of your ovule. You have a micropylar end, you have a chalazal end, and then you have the hilum, the point of attachment where it is attached, the ovule is attached to your funicle. Okay. What would be the heart rate of a person if the cardiac output is 5 liters? So, cardiac output is 5 liters. We all have approximately 5 liters of blood. The blood volume in the ventricles at the end of the diastole is 100 ml. And at the end of the ventricular systole is 50 ml. So, at the end of the diastole, it is 100. So when it opens up the ventricles, like when they are what you call as what do you call that, diastole is like relaxation. So when they are relaxing, it is 100. When they are contracting, it is 50. That means how much is going out is 50. So you have 100 right at the end of the diastole, you have 50 at the end of the systole. So you have a stroke volume, you can say, of around 50. So the blood which is pumped out, that is around 50 ml, right? So the stroke volume which is there, it is around 50 ml. So stroke volume is 50 ml. Your cardiac output, we know, is heart rate into your stroke volume, right? So cardiac output is how much? 5 liters. So 5 liters is how many ml? 5000, right? So you have 5000 ml. So cardiac output is 5000 ml. We want what is called as the heart rate. So this heart rate we want to find out, and stroke volume we just calculated. Stroke volume was 50. So stroke volume is 50. We want to know the heart rate. So heart rate is 5000 divided by 50, right? So 5000 divided by 50 will give me how much? Around 100, right? So my what I was calculating, the heart rate, right? So heart rate is 100. So we have 100 here. Yes, fourth option. Yes. So fourth option here is 100 beats per minute. It's really beating fast, because normal heartbeat, we know how much it is in 1 minute. It beats how many times? 72, right? So this is having a increased heartbeat. Maybe उसने कोई कुत्ता बिल्ली देख लिया होगा, या आजकल हमारे इधर वो बिपट्या ज्यादा आ रहे हैं, तो वो देख लिया होगा, तो उसका हार्टबीट कितना हो गया होगा? 100, right? So anyways, so cardiac output, heart rate into stroke volume. I hope you got how you got the stroke volume. At the end of the diastole, the blood was 100. At the end of the systole, it was 50. So how much was given out was 50. So jitna given out utna stroke volume. So cardiac output was 5000. So I calculated this. Okay, conversion of glucose to glucose 6 phosphate. Yeah, so this is the first reaction in what you call as glycolysis, the first step in your respiration, right? That is glycolysis, the EMP pathway. It is catalyzed by which enzyme? We know that many enzymes are there. You're lucky that your NCRT doesn't have all the enzymes. So probably only enzyme which is mentioned in your NCRT, out of all these, NCRT mentions only hexokinase, right? So hexo is a hexose sugar that is containing 6 carbons, and kinase, it is adding what is called as your ATP. So the first irreversible reaction where your ATP is donating a phosphate and it is converting your glucose to glucose 6 phosphate, it is being catalyzed by your hexokinase. These are not mentioned, but just to tell you that I am intelligent. So phosphofructokinase, we know that it is responsible for converting fructose 6 phosphate to fructose 1,6 bisphosphate. Aldolase is the enzyme which cleaves it, which cleaves your fructose 1,6 bisphosphate to your dihydroxyacetone phosphate and your phosphoglyceraldehyde, what you call. And then enolase, has to do something with water. So that we'll discuss some other day, or you can watch the video on glycolysis, which I'll be giving in the description box. So these are some of the questions of your NEET. You are going to tell me the year when were these questions asked. So stay tuned with me, Professor Girish Kukreja, for more in biology. Thank you.